Thank you for allowing me to join you in this seminar presentation. It's great to be here talking about Lake Hazen and the work that I've gotten to be a part of. Many of you know this lake very well, so it's lovely to be speaking to you about something I know that many of you are passionate about, and presenting some of your own work back to you. So just a brief outline of my talk today. I will start with the temporal trends of mercury in fish generally, and its relationship to the temporal trends in atmospheric mercury. I will briefly touch on the char monitoring project, which is a long-term study of trends in mercury and other contaminants as well in Arctic char. Then I will talk a lot about Lake Hazen and why it is a unique and valuable study site at the top of the world. It is home to two char, so there's one species of fish, Arctic char, and there's two morphotypes of char in Lake Hazen. So we'll go into more of what that means as the talk moves forward. Our approach for this study was developing a conceptual model to explain trends in mercury concentration in these two morphotypes of Arctic char. And we incorporated climate variables as well as mercury sources, which I'll get into that soon. And then we developed statistical models based on our conceptual models to really hammer out the influence of the climate variables and the different sources of mercury to the char. We also just briefly compare trends in mercury in the char and Hazen with Arctic char and some of the other char monitoring project lakes. And then there are some future work ideas and recommendations. So some of you might be familiar with this figure. It's been around, I think, since 2009, but it was updated in 2019. And this is just kind of a conceptual diagram of the relationship between air mercury concentrations, so atmospheric mercury concentrations, and the concentrations in biota. And as you can see, in 1850 or so, humans really began increasing the mercury concentrations in the atmosphere. And then we saw a steady incline as well in the biota. After that, things get a little bit more chaotic. And so we get some noise in the signal, some feedbacks going on in the biotic mercury that we don't see coming directly from the atmosphere. So a little bit of disconnect between the air mercury concentrations and the biota concentrations. However, and nowadays we have implemented some controls and we're reducing our air mercury emissions, but we are still in this period where there's a lot of disconnect between the air concentrations and the biota concentrations. So there's a lot of noise in the signal, if you will, but that noise is actually valuable noise in a way because we can learn a lot about ecosystem processes by examining that noise. So just to get into, just to outline this entire process and get us all kind of thinking along the same lines, you all know that there are a lot of abiotic processes, and Kasia has just walked us through some that I didn't include on this slide, so I'm glad that she presented before me. So here I'm showing the atmospheric transport of mercury to the Arctic. And after that has occurred, mercury moves around quite a bit across the landscape. There's emissions and re-emissions, and there's now legacy mercury, and there's deposition across the different areas of the landscape, and that can be affected by proximity to the coast and many other complex processes with the mercury chemistry in the atmosphere. So we've got, including the effect of the polar sunrise and the big deposition events that occur that have been very well studied right outside of Lake Hazen at the alert station. So all of these complexities are part of our signal. And then we get into the biotic processes. So for mercury, our first step in the mercury that eventually we monitor in the char is methylation, which occurs in the sediments of aquatic environments. And then we have bioaccumulation, which is the increase in mercury in an individual organism over time. And then we have biomagnification, which is the phenomenon of mercury being in very high concentrations in our predators, especially our apex predators, relative to their prey items. So we see the apex predators tending to have the highest concentrations of mercury due to bioaccumulation and biomagnification. So I wanted to go over why Lake Hazen research is particularly relevant for the AMAP program. This study in particular addresses aspects of the very first overarching policy relevant question in the latest AMAP assessment. What are the human and environmental drivers of mercury levels in the Arctic? This also speaks to the Minamata Convention effectiveness evaluation, because one of the main struggles in the effectiveness evaluation process is linking the biotic compartment and the abiotic compartments of mercury, and seeing how our reduced emissions are actually having effects in relevant biological compartments. And so this study is an example of the type that is called for in some of the Minamata Convention effectiveness evaluation documentation for cross-media data, which incorporates modeling and a top-down approach to understanding the relationship between the different compartments. So, some fun facts about Lake Hazen. Lake Hazen is the largest lake north of the Arctic Circle when you consider it by volume. It is in the bowl of a thermal oasis within a polar desert type of landscape. And it is, as you can see in the map here, fed by glaciers. So again, thank you for the setup, Kasha. Summer temps in this basin can reach up to 20 degrees C, so it really is a thermal oasis. We see artifacts indicating that the Independence 1 and 2 cultures from Greenland were active in this watershed. And later, the Hazen Valley was used by the Thule people, who are ancestors of the modern Inuit, and they migrated all the way from the eastern Arctic, so Alaska, as well as seasonally exploited by modern Inuit as well. So Arctic char, again, are the only fish in Lake Hazen, and there are two morphs, a larger pelagic cannibalistic morph and a smaller benthic insectivorous morph. And we'll be looking at both of those in depth today. The food web is quite simple, based on diatoms, and then chironomids and char. And then there's a bit of pelagic zooplankton as well, playing a minor role in the fish's diet. Lake Hazen has its own website as well. I just wanted to point that out and show you a great picture of this lake and how gorgeous it is. And again, this is where I got the top of the world little phrase. Some more fun facts about Lake Hazen. The climate warming in this lake has resulted in a 4-fold increase in glacial meltwater to the lake for the period of 2008 to 2012. And the average ice-free area during the growing season has increased about 40% from the early 2000s to around 2020. We also see a shift in diatom assemblages responding to decreasing duration of lake ice and snow cover, and higher fluxes of mercury to the lake sediments due to higher particle sedimentation rates during this time period. So here we can see a figure showing the modeled annual glacier runoff and how that's corresponding to mercury deposition in the lake sediments. So this is relevant for us because we're interested in how all of this warming and all of these phenomenon that we're seeing around Lake Hazen have impacted the trends in mercury in our Arctic char. Oh, and I should mention that they are landlocked Arctic char, so they're not migrating to the sea. So here is a little bit about how these char were collected. The collections began back in the 1990s, and collections were carried out both by gill netting and by jigging through the ice, as you can see in the photos. And then this is how the samples were handled. They were stored on ice and dissected quickly after harvest. And then mercury and stable isotopes were measured at in the labs at the lower latitudes. So our first task with this data was to categorize which morph are our char falling into. So we used a clustering analysis on our delta 15N by fork length relationship for these fish. And we did find that they really cluster nicely into two clusters. So the upper cluster is that cannibalistic type, and the lower cluster is the insectivorous. And then our next task, as anyone who works with fish mercury is familiar with, you've now got to reduce a little bit of your variation. That's due to the size of the fish and the age of the fish, as we touched on in our earlier slides, to get a good trend line. So we don't want our trends to be influenced by the fact that maybe we caught extra large fish in a certain year or something like that. So we went through a process of removing some of that variation, and then we used the same methodology that the last AMAP report used to generate our trend lines and look for change over time. So we use these generalized additive models. And as you can see, for both the large form and the small form char, we see a significant decreasing trend in mercury concentrations. So now we're ready to get into the inclusion of environmental variables. So for Lake Hazen, because it is so close to the alert station, which is a station for atmospheric mercury modeling as well as a weather station, we had a lot of data available to us to incorporate into our models for fish mercury concentrations. So we took advantage of the total gaseous mercury concentrations and that weather data, and we broke that into different seasons. So we followed in the footsteps of Dr. Steffen and broke the data set into seasons based on spring, summer, winter, and fall. We then offset the fall and winter because, as you saw in the previous slide, the char sampling always occurred in summer. So we subtracted a year from our fall and winter seasons and lined them up that way in our analysis. So again, the weather data that we got was rain, snowfall, snow depth, and temperature. We also used some of the glacier runoff and mass data that is kind of updated from a previous study, which from the Gilman Glacier, so how much runoff is coming into Lake Hazen. And then we added in the ice cover phenology for Lake Hazen. So the date of freeze up, the date of breakup, and the open water area during the summer. Additionally, we searched for oscillations, which those are those pressure variations that kind of control weather patterns over large spatial areas and sometimes over large time periods. So for Lake Hazen, we found that the Arctic oscillation and the Arctic multi-decadal oscillation, so those are similar oscillations that occur on different time periods, were selected as the most influential for this area. And so those are our environmental variables that we had to use to model first growth and then mercury concentration in both morphotypes of our Arctic char. So this is kind of like a schematic of our modeling process. So in our first step, as you can see in the upper model, is modeling all of the variables that have a direct connection to Arctic char growth. We use length at age as a proxy for growth. So that was our growing season variables, so our summer rain, summer air temperature, and the phenology of the lake ice. And then we did a stepwise modeling approach. So we modeled these direct variables first. Then we ran a separate model where we added in the climate oscillations. So that was our growth model. And then for our Mercury model, we had the Mercury sources, so the glacier runoff and the atmospheric mercury concentration. Also, we know that snow can contribute to mercury levels in lakes. So we looked at that to see if that was a potential source for the mercury in the Arctic char. And then we did the stepwise thing again, where we added in the oscillations in a second run of the model selection process. So this, now we'll kind of look at the results of these models. So I have the schematic here on this slide, and this is our growth model. And in the table below the schematic, you can see that we ran this model for each of our morphotypes, the large and the small. And the statistics over on the right side are our log likelihood and our ICOCI information criteria, and the change in that criteria for the models. So this was like we ran every possible model that did not have correlated covariates and selected for the ones that were most predictive for each of our morphotypes. So for our large morphotype, we just have the one. And as you can see, I will walk you through how to read this table as well. So I'm adding some circles and X's up on this schematic. So the green indicates a positive relationship. So an increase in summer rain leading to an increase in char growth. And we see that also for the ice-free area in the summer. And so then we see the red circle is indicating that later freeze up had a negative effect on the char growth. So that's what that means. And then we did not see that summer air temperature or the oscillations had an effect on these models at all. So when we did add in the oscillations, that didn't add any significance to our model, so we can leave that off. But yes, again, just to summarize, we see that the later freeze up leading to less growth, and we see summer rain having a positive effect on growth for both of our morphs, and ice covered area having a positive effect for the large morph, the pelagic morph. So now let's get into our Mercury model results. And as you can see, this is our first step. So we don't have our oscillations in this model. So in this model, we started with 14 variables. And again, we went through the selection process to find the most significant predictors for the large and small morphs. And we found that first of all, we found that the fall and winter seasons were the most, were incorporated in these models, but spring and summer were not. We also see that glacial runoff and seasonal snow, which we included to be potential sources of mercury, were both in the models, but they were negatively related to char concentrations. Then we see that our atmospheric mercury was positively related. So we're seeing fall and winter atmospheric mercury being linked to higher concentrations in our large morphotype char. And again, that was only for our fall and winter seasons. So that was good information. We took that information into account when we moved into adding in the oscillations to these models. And we used only the fall and winter oscillations because that was the season in which we were seeing the effects on the chart. So that's where the stepwise part of this comes into it. And again, walking through connecting the table to the conceptual model with these circles and X's. Once we added in the oscillations, the glacier runoff did not come up as significant in our models at this point. However, we still see the atmospheric mercury being important for the large morph and being positively related to char mercury concentration. We see seasonal snow staying in for this small morph only, and that being negatively related to mercury concentrations in that morph. And we did see a positive effect of including our climate oscillations with, as you can see, that fall AMO index for the large morph being the highest coefficient of any of our coefficients in any of our models. So I should have mentioned too that all the environmental data was scaled prior to the modeling process. So when you're looking at the coefficients in these tables, you can interpret them as you would interpret effect size because of the scaling that went into it. So a large effect size of the fall AMO on the large morphotype char. Okay, so that's a lot of statistics, and the statistical analyses can only take us so far. And then we need to come back into real life and kind of find out what are the mechanisms and processes that are potentially leading to the effects that we're seeing in the statistical models. So here we see, just recovering our results a little bit, the strongest predictor of large char growth was growing season ice cover. So more ice led to more growth for the large char. Increasing the flow into Lake Hazen could actually reduce mercury bioavailability by increasing the outflow volume or burial into the profundal zone if mercury is particulate bound. So I think that is my logic for why our snow and our glacier output were negative coefficients. They were significant, but they were negative for predicting char mercury. So I'm looking at those possible mechanisms for explaining that. Again, we see that mercury concentration in the small benthic feeding char was negatively linked to fall season snow. And in the growth model, that was weakly linked to later freeze up. So the fall season appears particularly important to that small benthic feeding morph. Then surprisingly, despite the large interannual variations in the glacier runoffs, we don't see that relating to char mercury or char stable isotope profiles. And again, to explain the oscillations, we know that high values of both the AMO and the AO are associated with more mixing. So warmer Arctic temperatures and a weaker polar vortex, conditions that promote mixing of air masses from lower latitudes, particularly Eurasia. And we also know that that is a big source of mercury to the Arctic atmosphere, particularly in the winter season. So that may be an explanation for what we're seeing in the chart. And then again, to hit on the Arctic's Great Lake, as discussed in the introduction, studies that link mercury concentration in the atmospheric compartment or other abiotic compartments to mercury concentrations in biota are quite rare, but they are quite valuable as well for effectiveness assessments. So again, just Lake Hazen's unique position in the Northern Hemisphere, the research station and the alert station that's there make this lake an excellent site for mechanistic studies resulting from this type of top-down approach. So just to wrap up the future work and recommendations, every finding of our models is an opportunity for a mechanistic investigation somewhere down the line. So it just brings to mind a plethora of questions that we can answer or try to. But I would say one of our untapped resources that could really answer some of these questions potentially, or maybe come up with better questions, is the traditional knowledge about Lake Hazen, which I am not aware that there's a whole lot of that being used by the scientists at the moment. Maybe some of you all can educate me on that. Another great potential way of investigating these is the mercury isotopes that could really get us to more of a fingerprint of where the mercury in the char is coming from, what source it's coming from, and how it moves through the system. And another big unknown is the chironomids. They have, to my knowledge, the chironomids in this lake have not been studied for quite some time, and how their mercury concentrations and how those are being affected by climate change is probably the #1 way that the char trends are being influenced. So those are all kind of little branches off of this research. And just wanted to let you also know that we see length at age increasing in several small Arctic lakes that are also under the umbrella of the Char Monitoring Project, but not in the Lake Hazen char. So char are growing faster, and this could be an effect of enhanced growing season. So warmer summer temperatures leading to increases in primary productivity and warmer temperatures. And that is in some of the other sites in the high Arctic under the umbrella of the Char Monitoring Project. So with that, I would like to thank my co-authors for this opportunity to put a few brush strokes on their amazing portrait of Lake Hazen, and also to you webinar organizers for allowing us to share our research with you today. And of course, our funding agencies and those from Parks Canada who helped collect these samples. So with that, I will leave off.